Welcome back, dear students. Today I shall be discussing the second part of the physiology of stomach, that is the secretory and the digestive activities that is going on inside the stomach. So without much ado, let's continue our discussion. The secretory events of the stomach. Before we go into the details of the secretory events, it would be better if we discuss the glands that are responsible for gastric secretion in a nutshell. So what are the glands that are responsible for gastric secretion? The glands are simply known as gastric glands, and the mucous membrane of the stomach or the gastric mucosa contains several gastric glands, and according to the location of those glands in different parts of the stomach, they are classified into three main types. That is the cardiac gastric glands, that is the gastric glands located in the cardiac region of the stomach. The fundic and body gastric glands, that is the glands located on the fundus and the body of the stomach, and the pylori glands. So the gastric glands are all distributed throughout the stomach. Their characteristics differ on the basis of where in the stomach they are located. And what is the difference? The difference is simple. It has been found that the cardiac and the pylori glands, they secrete mainly mucus, water and bicarbonate. They hardly secrete any acid or pepsin. While the fundic and the body glands, they are much more evolved, much more larger and complex, and they secrete the acid peptic juice. So cardiac and pylori glands are simple glands. Often there are only goblet cells, no glands per se, and in those regions the gastric secretion is mainly of mucus, water and bicarbonate, while in the fundic and the body regions the glands are much more well developed, deep seated, and has variety of cells, each variety of cell secreting a different type of compound, some secreting the acid and the Cassell's intrinsic factor, some secreting pepsinogen, so on and so forth. Now a typical acid secreting gland is straight and tubular in cross section with several types of cells lining its walls, and they are of different types. One variety are the isthmic cells, that is the cells just lying below the surface near the area where the surface pit connects with the gland's tubular lumen. That area, the cells lining those areas are known as the isthmic cells, and they secrete mainly mucin. Then below them are the neck cells or the regenerative cells or the stem cells, which can migrate in both directions and give rise to other variety of cells. Can be the mucin secreting cells, can be the peptic cells, can be parietal cells, or can be the enterochromaffin like cells. So the neck cells are the regenerative stem cells. Thereafter the glands are lined by different varieties of cells, namely the chief cells or peptic cells that secrete the proenzyme pepsinogen. Then the oxyntic cells, also known as the parietal cells, they secrete the hydrochloric acid as well as the intrinsic factor of Cassell. And also another variety of cells are there which are known as the enterochromaffin like cells or ECL cells. They secrete histamine. These are the main types of cells lining a gastric gland. This is a schematic diagram of a single gastric gland in cross section. You can see it is long, straight with a tubular lumen. On the surface, you can appreciate there are surface mucus cells which somewhat invaginate in the upper parts of the tubular lumen and give rise to the isthmic cells. These cells are mainly responsible for secretion of mucus, bicarbonates, water, and also a peptide known as a trefoil peptide, which is important for the formation of the gastric mucosal barrier, which helps to protect the gastric mucus membrane from autodigestion by its own acid and pepsinogen. Just below the isthmic cells, you can find the neck cells, which are the stem cell compartments. These cells, as you can see in the diagram shown on the left hand side, it is shown cell migration can occur in both directions. They can give rise to the mucus cells as well as different varieties of other cells like parietal cells or chief cells. If they migrate above or upwards, they will give rise to the mucus cells, and if they migrate downwards, they will give rise to the special varieties of parietal and chief cells. Then obviously comes the parietal cells. You can see the lumen of the cells open into the tubular lumen of the gland. If you look closely, greenish colored cells are shown, which itself has small lumens on its luminal surface, which is opening into the gap of the gastric gland, and from there the acid and the intrinsic factor are secreted. Thereafter, you can find the labeling as the ECL or enterochromaffin like cells, which secrete the compound histamine. You can appreciate that these cells do not open into the lumen. They are more opening towards the basolateral side. That is because their secretion, that is histamine, is not required in the gastric lumen. It has no job there. It is required for its action on the parietal cells for their secretion. So they are emptying their contents in the blood, or rather the extracellular fluid surrounding the parietal cells rather than the lumen of the gastric gland. Thereafter comes the chief cells or pepsinogen secreting cells. Obviously they are secreting pepsinogen, which is a zymogen or a proenzyme, which on reaching the gastric gland's tubular lumen and surface is activated to its active enzyme pepsin, which breaks down bonds between amino acids in the food product. All these secretions come out through the lumen of the gastric gland, the tubular lumen, through the gastric pit. You can see the arrow showing acid, intrinsic factor, pepsinogen all coming out, reaching the surface of the stomach, where it can act on the ingested food particles. Composition of gastric juice. By gastric juice, it is meant that all the secretions of all the gastric glands taken together, means the secretions from cardiac, fundic, body as well as pylori glands taken together, and not only the acid secreting glands. So gastric juice is the combination of the secretions of all varieties of gastric glands. What is the volume of gastric juice produced per day? On an average, it is 2 to 2.5 liters per day in an adult. The pH of gastric juice is very acidic. It varies between 1 and 2. When food is there in the stomach, it is more acidic. When the stomach is empty, it is less acidic. That is, it is more towards 1 in a fed state and more towards 2 in a empty state. But in toto, it is always very, very acidic. What is the constituents as per water and solids? 99.45% is water and only 0.55% is solids in gastric juice. So you can imagine there is a lot of water. The main constituents of the gastric juice. First of all is the hydrochloric acid or HCl. The parietal cells or the oxyntic cells are the ones that produce HCl. They secrete H plus and Cl minus separately, which combine inside the lumen of the gastric gland to form the HCl. Normal concentration inside the gastric lumen. Here the gastric lumen means the surface of the stomach, not the lumen of the gastric gland. On the surface of the stomach, the normal concentration of hydrochloric acid is between 40 to 60 milliequivalent per liter. But if we have taken a very heavy meal or a meal which is very spicy, loaded with fats and proteins, the levels maximal concentration can reach up to 160 milliequivalent per liter. Next important constituent of gastric juice is the proenzyme pepsinogen. It is secreted by the chief cells and converted to the active form firstly by gastric acid and then by autocatalysis. Means some amount of pepsinogen which comes out initially is converted to pepsin by the acid. Then the pepsin itself can convert more pepsinogen to pepsin. So initially by gastric acid and then by autocatalysis. This pepsin, it is a endopeptidase, means breaks peptide bonds between two amino acids in between a amino acid chain, not at the terminal points. And it preferentially breaks the bonds between aromatic amino acids. So it breaks down large protein molecules into small proteoses and peptones. And it has been quantitatively found that it is used to digest about 20% of the ingested proteins. So only 20% of ingested proteins can be digested by pepsin alone, while the rest will be digested only after it reaches the small intestine. So pepsin is an important proteolytic enzyme, but not the only enzyme. The next important substance that is secreted by the gastric glands and an important component of gastric juice is the intrinsic factor of Castle, or simply intrinsic factor. What is this intrinsic factor? Chemically, it is a glycoprotein secreted by the oxyntic cells along with the hydrochloric acid. So the two things that are secreted by the parietal or oxyntic cells are the acid, hydrochloric acid, and the intrinsic factor. What is the job of this intrinsic factor? This intrinsic factor is important for absorption of vitamin B12 in the diet. Though this particular glycoprotein is secreted from the stomach, it does not bind to the vitamin B12 until and unless both of them reaches the small intestine, namely the duodenum of the small intestine. Only in the duodenum does the combination between the intrinsic factor and vitamin B12 occurs. Prior to that, the vitamin B12 that is liberated in the stomach due to the action of the acid on the food is protected from stomach's acidity by a special protein known as haptocorrin. This haptocorrin is secreted mainly from the saliva. So the salivary haptocorrin passes along with the food bolus into the stomach It is resistant to acid peptic digestion. Once the cobalamin, that is the vitamin B12, the other name of vitamin B12 is cobalamin, it is released from the diet, the haptocorrin binds to the cobalamin and protects it from the acidity of the stomach. Next, the cobalamin haptocorrin complex reaches the duodenum, and the intrinsic factor also has reached the duodenum because it has been secreted by the stomach. In the duodenum, there are proteolytic enzymes that degrade the haptocorrin and liberate the cobalamin. Now the cobalamin binds to the intrinsic factor and then it travels down, reaches the terminal ileum, where there are receptors specific for the intrinsic factor, and cobalamin is internalized into the cells by receptor mediated endocytosis. So that is the role of intrinsic factor in vitamin B12 absorption. Deficiency of intrinsic factor leads to a condition which is known as pernicious anemia. The word pernicious means something which is very difficult to treat. Why this nomenclature? Because in olden times, when mechanisms were not known, whenever a person suffered from anemia, he or she was given iron. Now if the cause was due to intrinsic factor deficiency, which was not known at those times to the physicians, they found that even if we give iron, the anemia is not being corrected. That is why they termed the disease as pernicious. That is, even after treatment, the anemia persists and it requires very vigorous treatment. Vigorous treatment in the form that lot of vitamins also had to be given along with. Now this form of anemia also didn't properly respond to vitamins. At that time it was not known which vitamin, so lot of vitamin complexes were prepared and given, but the patients with this form of anemia either didn't respond or responded partially. That is because the parietal cells of these patients were already destroyed due to the autoimmune disorders. There was autoantibodies against the membrane of the parietal cell in these patients. So the parietal cells got destroyed. There was achlorhydria or hypochlorhydria, and as well as deficiency of intrinsic factor. So you can imagine the cobalamin in the diet was not getting released in the stomach due to deficiency of the acid first of all, and if at all there was some acid, the cobalamin which was released and carried down by haptocorrin wasn't taken up because of the deficiency of the intrinsic factor. So even if you give orally vitamin B12, these patients will not respond. And so after years and years of experiments, it was found that certain patients of anemia, they require injectable vitamin B12 for the anemia to be corrected. So the vitamin B12 was named the extrinsic factor, and there was some intrinsic factor that was proposed to be present, which was responsible for absorption, because without it, even if we give vitamin B12 orally, vitamin B12 was not getting inside the body and anemia was not corrected. So even though vitamin B12 is there in the diet, iron is there in the diet, the person is still suffering from anemia. So it was a very pernicious condition. That is why the name pernicious anemia and the importance of intrinsic factor. The next constituent is the gastric mucus itself. Why it is important? Because it is an important contributor to the gastric mucosal barrier. That is the barrier that protects the gastric mucous membrane from autodigestion by its acid peptic juice. Now this gastric mucus not only protects the stomach from its autodigestion, but also helps to give a buffer against physical trauma by solid food. There are two main types of gastric mucus, one that is visible and one which is soluble in the visible mucus. Means ones that can be visualized and one which is soluble inside that visible mucus. What is the visible mucus? The visible mucus is the secretions of the surface epithelial cells of the gastric mucosa, and it contains large amount of water and bicarbonate. It is alkaline and helps to form the main mucosal barrier that protects the stomach wall from autodigestion. It sort of forms a layer between the highly acidic gastric luminal juice and the luminal surface of the epithelial cells. So there is a alkaline layer between the highly acidic contents of the stomach and the mucous membrane of the stomach, or the luminal side of the plasma membrane of the surface mucosal cells. This visible mucus, it is secreted all throughout the day, even during resting state, and rate of secretion increases with food intake. The soluble part of the mucus are the different proteins, and they are mainly secreted by the isthmic cells of the gastric glands, and their secretion has been found to increase with food intake and comes down when the stomach is at rest. It contains one protein which is mucin. It is a tetrameric glycoprotein, also another form of peptide known as trefoil peptide. All these mucin and peptides, they give the gel sort of character to the mucus and also helps to trap the bicarbonates inside the mucus so that they do not get blown away with the movements of the stomach and remains near the surface of the cells, so that they can neutralize any H+ that leaks towards the surface of the mucosal cells. The next constituents are the organic matters, mainly the enzymes like gastric lipase, gelatinase, lysozyme, carbonic anhydrase. Among them, gastric lipase, as the name suggests, is involved in fat digestion, though it is not a very important enzyme as such. Gelatinase and lysozyme are the enzymes that are more involved in the innate immunity part of stomach. That is, it helps to break down the cell walls and protective capsules of potential pathogenic organisms that we take along with our food. And carbonic anhydrase is an enzyme which is involved in the HCl secretion. Though it is mostly present inside the parietal cells, some are secreted also. Next are the inorganic constituents. Most important obviously is the H+ and the chloride minus ions that forms the hydrochloric acid. Other important anions and cations are the sodium, potassium, magnesium, phosphate and sulfates. The next topic of discussion is mechanism of HCl or hydrochloric acid secretion by the parietal cells of the stomach. As I have said previously, the HCl or hydrochloric acid is not secreted as a single compound but as H plus and Cl minus ions separately by the parietal cells. The enzyme that is involved in the secretion of H plus ions is the cardinal enzyme, and it actively transports the H plus from the parietal cells into the lumen of the gastric glands. Its name is the H plus K plus ATPase or hydrogen potassium ATPase, but more commonly it is known as the proton pump. Why a proton pump? Since it pumps out one H plus, which is basically proton, from the parietal cell into the gastric lumen. So the proton pump is the cardinal enzyme involved in H plus ion secretion by the parietal cell. Why do we need a pump? The explanation is obvious. Since H plus ions are in much more concentration outside, that is in the gastric lumen, compared to the inside of the parietal cells, hence we have to use energy by breaking down ATP through a ATPase, and we have to pump out the H plus. And since we are pumping out one positively charged ion, to maintain the electroneutrality, one potassium ion comes in. So that is the mechanism of hydrogen potassium ATPase or proton pump. It pumps out one H plus and takes in one K plus. This diagram is slightly fuzzy, but it depicts the steps in the secretion of H plus and chloride ions by the parietal cells. You can see parietal cell is written at the top of the diagram, and the luminal and the basolateral or plasma sides of the cell is marked. The first step is the carbon dioxide, which is abundantly produced due to cellular metabolism. Maybe the metabolism occurred within the parietal cell or some other cell outside. The carbon dioxide, being soluble in plasma membranes, lipid soluble, easily diffuses in, and once it diffuses inside the parietal cell, due to the presence of the carbonic anhydrase enzyme, this enzyme leads to the combination of CO2 with cellular water and formation of the carbonic acid or H2CO3. This carbonic acid is a very unstable compound. Immediately after its formation, it breaks down into H plus ions and bicarbonate ions or HCO3 minus ions. So the carbon dioxide which has entered the parietal cell has got converted into bicarbonate and H plus in combination with cellular water. Next, the H plus that is formed due to the action of the carbonic anhydrase is pumped out by the proton pump, and one K plus is taken in. So the H plus ion is now in the lumen of the gastric gland. The K plus ion that has been taken in by the proton pump immediately goes out because there are lot of potassium channels that lines the luminal side of the parietal cells. Potassium was already in excess inside the cell because intracellular fluid contains lot of potassium. So whenever a potassium comes in, it has the tendency to go out because of the concentration gradient, which is maintained by the sodium potassium ATPase that is present in all cells in the basolateral side. In the picture also you can see in the lower down in the picture you will find a sodium potassium ATPase is there that is pumping out sodium and taking in potassium continuously and keeping a high concentration of potassium inside the cell already there. So the excess potassium that enters due to the H plus K plus ATPase immediately goes out through the open potassium channels which are located in the luminal side, and basically potassium is continuously recycled. It enters the cell and goes out through the channel and again back into the lumen of the gastric glands. Next, the bicarbonate which is formed due to the action of the carbonic anhydrase is accumulating inside the cell, and now it just goes out of the cell because of its high concentration through a exchanger protein which is known as the bicarbonate chloride anion exchanger protein present on the basolateral side. This bicarbonate goes out and in exchange of it chloride comes in because of this particular protein present on the basolateral side. The chloride that comes in is also excess because bicarbonate was excess, chloride is also excess. So to maintain the electroneutrality, that chloride also has to go out. Now the channels for chloride to go out is located mainly on the luminal side. The chloride ion that accumulates because of the exchange enters the gastric lumen through the chloride channel in the luminal membrane. So now both the H plus and Cl minus are now present in the luminal side of the gastric parietal cells, and now they can combine inside the lumen of the gastric gland to form HCl and do its job in digestion and innate immunity. Till now we have discussed the major constituents of the gastric juice, namely water, bicarbonate, hydrochloric acid, pepsinogen, intrinsic factor, mucus, and so on and so forth. And we have also discussed the different varieties of gastric glands that secrete the gastric juice. Also the mechanism inside the parietal cell that lets it secrete the H plus and chloride minus ions. But what is the regulation of the gastric secretions, or in other words, how gastric secretion occurs in relation to the different phases of feeding in a 24-hour period? As we can all say from experience that we are not always feeding. There are phases of feeding and then phases of fasting in a 24-hour period. How does gastric secretion differ during these different phases? So that is the topic of our next discussion, that is the phases of gastric secretion. First phase is the interdigestive phase. That is the phase when there is no food in the stomach and the main movement in the stomach are the migratory motor complexes. Does gastric secretion occur during that phase? Answer is yes. And what is the stimulus for gastric secretion or the secretion by the gastric glands? Who controls the secretion during that interdigestive phase? The answer is the parasympathetic tone from the vagus nerve. In our body, there is always a basal parasympathetic tone. and for the gastrointestinal tract, that tone is maintained by the fibers of the vagus nerve traveling down from the brain. During the interdigestive phase, whatever gastric secretion occurs, it is controlled by the basal parasympathetic tone, and the character of that secretion is mainly watery with bicarbonates. Very little acid and pepsin secretion occurs during the interdigestive phase. The next phase, or the phase after an interdigestive phase ends, or more properly, the phase that terminates the interdigestive phase, is the cephalic or cephalic phase. This phase occurs most commonly when food enters the mouth. So whenever a person starts eating, takes in the first bolus in his or her mouth, the interdigestive phase secretions starts getting terminated and the cephalic phase secretions begins. It has been found that during one proper meal, 30% of the total gastric secretion occurs even before the food enters the stomach, and that is due to the cephalic phase. It is also mediated by the vagus or the parasympathetic tone. The rate of secretion varies about 25 to 30 milliliter per hour, and it starts 30 minutes before food actually enters the stomach, or in other words, it takes about half an hour for the bolus to travel down from the mouth to the esophagus through peristaltic movements. As I have already said, the presence of food in the mouth is the most important trigger for the cephalic phase. However, sight of food, smell of food, or even thought of food can sometimes trigger the cephalic phase. The chemical mediators that is responsible for the cephalic phase are acetylcholine and gastrin releasing peptide, both of which are released from the vagal endings. The acetylcholine directly increases acid secretion by acting on the parietal cells, while gastrin releasing peptide, it stimulates G cells present in the stomach mucosa to secrete gastrin, which in turn stimulates acid secretion. The cephalic phase and the phase that follows it, that is the gastric phase, is somewhat overlapping. The cephalic phase begins when food is inside the mouth first time. It starts speaking, and once food enters the stomach, the secretion of the stomach is due to the gastric phase, but cephalic phase does not abruptly stop there. It has been found that it can continue up to 2 hours since starting of feeding. Or in other words, when a person takes in the first bolus, the cephalic phase begins. And if due to some reason he does not ingest the food, stops eating, then also once the phase has begun, it continues for about 2 hours and then stops. If the person continues eating and ultimately food reaches the stomach, the cephalic phase is taken over by the gastric phase. Otherwise, it stops there for after 2 hours. That is the significance of the line. So this phase terminates after about 2 hours from the starting point of feeding. The next phase after the cephalic phase is the gastric phase, and it has been qualitatively found that it accounts for 60% of the total gastric secretion during a certain meal. So 30% due to the cephalic phase and 60% due to the gastric phase. The trigger that starts the gastric phase is the entry of food into the stomach. So once the food bolus enters the stomach, the gastric phase will begin. Here the mechanism is both neural and local chemical mechanisms. Neural mechanism is simple, again due to the vagus nerve. As the food enters the stomach, there is receptive relaxation of the upper part of the stomach, which I have already discussed in my last class. This stretching of the stomach walls stimulates the vagus, and the vagus secretes acetylcholine that leads to secretion of the acid peptic juice. Also, this food product, after it enters the stomach, undergoes or starts undergoing digestion, and products of protein digestion due to the action of pepsin is now inside the stomach lumen. These products of protein digestion stimulate G cells present mainly in the antral mucosa or the mucosa of the gastric antrum, and the G cells, on being stimulated by the products of protein digestion, leads to release of the hormone gastrin. This gastrin again goes through the bloodstream, reaches the parietal cells. There is specific gastrin receptors are there, the CCKB receptors, through which gastrin act, and they cause increase in the acid peptic secretions. So gastric phase of secretion is due to entrance of food in the stomach. There is stretching of the stomach wall that stimulates the vagus nerve to secrete acetylcholine, and the products of protein digestion stimulates G cells to produce gastrin. This gastrin in turn increases the secretion of the gastric glands. The rate of secretion in gastric phase is much higher compared to the cephalic phase. It is almost double. It is 60 to 70 ml per hour. How long does the gastric phase continue? It continues as long as food is there in the stomach. That is the total gastric emptying time for a normal diet, which is approximately 3 to 4 hours. So once the food enters the stomach, the gastric phase begins, and it continues, or the stomach continues to secrete till the last food bolus has been eliminated from its lumen into the duodenum. The last phase of gastric secretion during one particular meal is known as the intestinal phase and accounts for about 10% of the total gastric secretion during a single particular meal. The trigger for the intestinal phase is the entrance of the products of protein digestion mainly into the duodenum and upper jejunum. These areas of the gastrointestinal tract also have the G cells, just like in the antral mucosa. These G cells, on being stimulated by the products of protein digestion, liberate gastrin that causes the secretion of acid peptic juice from the gastric mucosa. The rate of production of the acid peptic juice is much lesser compared to the gastric phase than in the intestinal phase. It is about 40 milliliter per hour. The intestinal phase is also marked by the release of inhibitory gastrointestinal hormones, namely secretin, CCK and GIP. These three local hormones, sometimes known as the gastrointestinal hormones, are produced by different cells located in the mucosa of the small intestine. They are secreted when either highly acidic food entering the duodenum or by the products of lipid digestion. They actually inhibit gastric secretion and contributes in terminating the gastric secretion after the food has left the stomach. So once we take food inside the mouth, the cephalic phase begins, which is neural in character, controlled by acetylcholine and gastrin releasing peptide released from the vagal ends. Then when the food enters the stomach, the gastric phase, which is the main phase of gastric secretion, begins. It is both neural and chemical in nature. The neural contributed by the vagus due to stomach stretching, and the chemical contributed by the gastrin liberated by the G cells of antral mucosa when they are stimulated by the products of protein digestion. As the food starts entering the duodenum from the stomach, the intestinal phase begins. The intestinal phase is purely chemical. The positive or reinforcer to the gastric secretion is the gastrin secreted by the G cells of the intestinal mucosa when they are stimulated by products of protein digestion, while the negative reinforcer are the hormones secretin, CCK and GIP produced by other cells from the intestinal mucosa when they are stimulated by the acidic chyme or the products of lipid digestion. A balance between the two continues as long as food is there in the stomach. Once the food leaves the stomach, the gastric phase terminates. Now the gastrin from the intestinal source is of much low power compared to the three hormones that is now trying to stop the gastric secretion, and slowly they get upper hand and the intestinal phase stops about an hour after the gastric phase terminates, and ultimately again the interdigestive phase takes over. So this is the gastric secretion phases in a nutshell. This schematic diagram tries to explain the regulation of acid secretion of the parietal cells. The parietal cells that secrete acid are under the influence of many chemical messengers. Among them, the three most important facilitating or the messengers that increase acid output are gastrin, acetylcholine and histamine. Source of gastrin are the G cells, source of acetylcholine are the vagal nerve endings, and source of histamine are the enterochromaffin like cells. They all act on the parietal cell to ultimately cause increase in acid production, while the main inhibitors are the hormone somatostatin, which is secreted by S cells located in the stomach mucosa, and prostaglandins, which are products of arachidonic acid metabolism present in the blood. They inhibit or decrease H plus secretion or acid secretion. Gastrin acts through the receptor known as CCKB. So gastrin has not got its own receptor. the receptor it uses to do its job is the receptor for cholecystokinin. So cholecystokinin has different types of receptors. One variety is known as CCKB. That is the one to which gastrin binds. On binding, a G protein which is known as GQ is stimulated, that causes activation of the enzyme phospholipase C. The phospholipase C enzyme ultimately causes increased amount of diacyl glycerol or DAG and inositol phosphate in the cytosol of the parietal cells. Diacyl glycerol ultimately leads to increased expression and activity of protein kinase C, which through different pathways ultimately leads to increased expression of H plus K plus ATPase pump on the luminal side of the parietal cells. More enzyme on the luminal side will cause more secretion of H plus. Also, the IP3 acts on the membrane of the endoplasmic reticulum and causes increased calcium concentration in the cytosol of the parietal cells. This increased calcium also helps in expression of increased amount of proton pump or H plus K plus ATPase enzyme on the luminal plasma membrane of the parietal cells. Next, acetylcholine, it acts through its receptor which is known as the M3 or muscarinic type 3 receptor. This muscarinic type 3 receptor can act by two ways. It can either increase the activity of GQ protein or itself can cause entry of calcium from the extracellular space into the parietal cell by opening up ligand gated channels. This can ultimately will cause increase in calcium concentration inside the cytosol and more expression of hydrogen potassium ATPase, or through GQ protein it will again go through the same pathway as which the gastrin acts. Next is the hormone histamine, the local hormone or the chemical, whatever you like to call it. It is liberated by the enterochromaffin like cells of the gastric glands. As I have already told, they secrete the histamine into the blood so that the histamine can go and act on the parietal cell basolateral membrane where special receptors known as the H2 receptors are present. This H2 receptor, after being stimulated by histamine, stimulates another type of G protein known as the GS protein. This GS protein causes activation of the enzyme adenylate cyclase that causes formation of the second messenger cyclic AMP. The cyclic AMP will go and cause increased activation and action of protein kinase A. This protein kinase A will ultimately through different steps lead to more expression of hydrogen potassium ATPase pump on the luminal membrane of the parietal cells. So ultimately all these three, gastrin, acetylcholine and histamine, will cause increased expression of the proton pump and that will ultimately lead to increased acid secretion. The somatostatin and prostaglandin, they decrease acid secretion because they act through another G protein known as the GI. Both of them have their own receptors, the prostaglandin having their own receptor, the somatostatin having its own receptor, but they will act through the G protein GI. This G protein GI actually inhibits or decreases the activity of adenylate cyclase. The decreased activity of adenylate cyclase leads to decreased production of cyclic AMP and decreased activity of protein kinase A. So the expression of the proton pump is decreased. From here, I would like to discuss a few points about certain drugs you have heard about. First is the drug pantoprazole or omeprazole or rabeprazole. What are these drugs? These are the drugs that we take to decrease acid production. How do they act? They act through their action on the proton pump itself, that is the H plus K plus ATPase. They inhibit the action of the H plus K plus ATPase. So they are very strong, because from the diagram you can easily make out the final path of acid production is the H plus K plus ATPase. So if you can inhibit that, acid production will go down. So that is why these drugs are also known as proton pump inhibitors or PPI. These are the mainstay drugs nowadays to fight acid peptic disease, which is a disease where there is increased production of acid and pepsin and ultimately gives rise to gastric ulcerations. So to combat that, we use the proton pump inhibitors. Another group of drug which is quite common and used to fight gastric acidity or excess acidity is ranitidine and famotidine. What are these drugs? These are the drugs that inhibit the receptor H2, means the H2 receptor of histamine. So they are the antihistaminic drugs which are specific for the H2 receptor, so sometimes also known as the H2 receptor blockers. So they will only inhibit that amount of acid production that is mediated through histamine. They will not inhibit the acid production through acetylcholine or gastrin. So they are less potent compared to the proton pump inhibitors. Other variety of inhibitors for acid production which are not used that much are obviously the inhibitors for the M3 receptors. They are the pyrenzepine and telenzepine. They are anticholinergic drugs. But since acetylcholine is required for various actions in different parts of the body, anticholinergic drugs just for decreasing acid production is rarely used or at all not used clinically, because they will give lot of side effects. So the main drugs for acid regulation or decrease in acid production are the proton pump inhibitors and the H2 receptor blockers. Gastrin receptor blockers have very little role. Mainly the advent of the PPI or proton pump inhibitors have made all types of anti-secretory drugs redundant, because if you can block the final step, there is no need to think about the previous steps. This is about regulation of acid secretion. I hope you have had a good idea about how the secretory events of stomach take place. And here I will stop. For any further question, you can ask me on WhatsApp in the student group you have. So thank you again, and we will meet again in the next class. Goodbye.